So the last part of the topic, okay, the fourth video is actually on a bit more of the basically point to control gene expression, coupling of RNA transcription and processing, plus a bit of translation and a comparison, side-by-side comparison of transcription in prokaryote and also in eukaryote. Okay, all the mechanisms, all the factors, and also some of the characteristics. So let's take a look at the coupling of RNA transcription and processing first. So we know that RNA polymerase 2 is the one again that supports coupling of transcription and pre-mRNA processing, and this both occurs in nucleus. So this is also governed by the work of CTD, which act as a landing pad, because CTD is actually sets of sequences on the DNA sequence. This is to enhance your mRNA maturation. So apart from that, CTD also stimulates capping, splicing, and of course polymorylation processes, and this has been proven in vitro in the lab. So this is the structure of the CTD of polymerase 2. So it has a stretch of 7 amino acid repeats in the C terminus, and the repeats are the YSPT SPS, so it actually codes for specific amino acids. You can look it up if you're interested to know what is Y, what is S, and then what is P. So for example, okay, the number of the CTD basically varies from one eukaryote to another. In yeast, there are 26 repeats. In human, there are about 50 repeats. And CTD gets phosphorylated at serine and tyrosine residue after a transcription initiation. So this is important, okay, for promoter to escape, okay, and also for elongation and post-transcriptional events. So apart from that, okay, apart from being the landing pad for different protein, CTD also is involved in elongation and post-transcriptional event. We have a very detailed mechanism of elongation, post-elongation, okay, and that's like basically elongation itself, okay, there's a lot of steps involved, which you will see in a few weeks' time. And the base of the CTDs is of course located near to the exit groove of the RNA. So basically this is a rough diagram showing how CTD act, okay, as the landing pad, as the start or as a place, okay, for the for example, for the A is for the capping mechanism, for the polyadenylation, for the transcription termination over here, for the spliceosome assembly, and also for the splicing activation, okay, to cut off your intron and to join your exons. In terms of RNA transport, we know that RNA could be transported, okay, between the cell compartments, but basically the types of RNA and how they are transported varies. So for example, you can see over here, all RNA could be transported from the nucleus to the cytoplasm, okay, in all cells. But okay, the tRNA could only be transported from the nucleus to the mitochondrion, and it could be located in many cells. For the mRNA, from the nerve cell to the oocyte, sometimes, especially in the fly embryogenesis. For the mRNA, for Dido, okay, it's actually from interior to posterior oocyte, and for plant phloem, usually for the mRNA is from cell to cell. And of course, there is a mechanism, okay, which govern the transport. So RNA, we know there are many types of RNA, as you can see over here, and we have covered it in the first two videos. So siRNA or small nuclear RNA basically helps, okay, with the splicing of RNA and also for the translation later on. tRNA, we know which is very crucial for the translation. mRNA, of course, okay, the powerhouse, okay, after transcription. rRNA, which is, okay, the housing for the translation, okay, and miRNA or microRNA, okay, of course it's not shown because it's actually micro, so it basically inhibits translation. So RNA has to be transported, okay, and the transport, okay, is whether it is exported or imported, okay, to and within the cells. So each RNA have different transport pathway through the NPC or nuclear pore complex. So mostly it would involve exporting pathway, okay, which is signal mediated by the nuclear export signal. So the next RNA binding protein would of course be needed to export all RNA except the tRNA because it has no adapter. So all these RNAs, okay, with the acceptance of mRNA, are dependent on a range GTP and also small res related GTPase. We will have a more detailed explanation on this, okay, in a few weeks' time. So GTPase enzyme will bind to the GTP to hydrolyze GTP. Okay, this is basically energy conversion to release energy. Now, I'll just move straight ahead again to the translation, because again this topic is just very general topic, a bit of an overview for gene expression in eukaryote. So translation in eukaryote involves initiation, elongation, and termination. So the mRNA would be translated to protein, of course, okay, as the end product via the ribosome and tRNA. So from the sequences again, of the mRNA, each trinucleotide codes for one amino acid. That much is known and always will be. So the final product is called a polypeptide, and it will form a protein depending on how it is folded. So this diagram depicts how translation occurs, and this is from transcription, whereby you get your mRNA. So hopefully it has been cut off from its intron and whatnot. So it has its poly A there, and hopefully with the cap. So everyone here, it goes through, or it passes through. So whether this is import or export, we will discuss that later. So basically, it will pass through the pore to go into the cytoplasm to be translated into protein. All right. So the last part of this topic is basically the differences between transcription in eukaryote and prokaryote. So I'm combining a few comparisons. You can actually find this all over Google, but I'm finding each and every one of them, even this one from journals, so that you can see there are more than what is being put in Google image. So these are all the differences that you will need to know, and you basically need to study, because I will usually ask this in final exam. So this is a bit more, but this is on the mechanism of translation. This one is basically on the transcription. This one is the differences in the translation. Even though the course name for MMBT 1153 is the gene regulation, mechanism of gene regulation, but translation is also something that comes after transcription. So sometimes this kind of differentiation question will be asked. So I would just like to summarize with this table for the regulation of gene expression in bacteria and prokaryote. In bacteria and eukaryote, the differences. So in terms of the level of regulation, chromatin modeling, and so forth, there is basically a very stark differences between them. As you can read here, transcription, we know for sure. RNA processing, yes, we know that in eukaryote it is very extensive. What I have shown you as an overview is basically just very general. But we will go into details. For the mRNA stability, some RNA for the prokaryote basically are documented, but basically for eukaryote RNA interference limits lifespan or the translation rate, and most of the time it would play havoc with the translation process and the protein produced. For the translation process, generally in prokaryote, regulatory protein would bind to mRNA and or ribosomes and affect the translation rate. But in eukaryote, regulatory proteins bind to the mRNA, ribosome, and also affect the translation rate, more or less similar mechanism. In the terms of PTM or post-translational modification, they basically have similar outlook, but with an exception of an additional step in eukaryote, whereby ubiquination target proteins for destruction by proteases, whereby this is basically not happening in bacteria. So there you have it, a general overview of gene expression in eukaryote. So next week we will go into detail of how genes could be regulated and the mechanism behind it.